recording. Great. Well, thank you all for being here today. This is our latest session of the VICC Virtual Molecular Tumor Board, and we're so happy to have Sumaiya Iqbal and Seulki Kwon from the Broad Institute to present on the Genomics to Proteins portal, which I've used in a couple of my variant interpretations. I find it really helpful when I'm right on edge, so I can't wait to hear more about it and to learn how to use this portal even better. So thank you so much for being here today and for sharing your expertise with us.
Go ahead. Yeah. Thank you, Beth. That was a great introduction. I will not take further time to introduce us, but I'll let Seulki to introduce herself real quick.
Hi, I'm Seulki. I'm a research scientist working with Sumaiya, and I do multiple bioinformatics and computational biology research with her. And this Genomics to Proteins portal is one of them, and we characterize mutations in terms of computational structural biology to help interpret unknown mutation in the context of disease. So I'm really excited to present. I'll start. I was waiting for you.
Yeah, no worries.
This Genomics to Proteins portal is a discovery portal to link genetic outputs to protein sequences and structures. And before start, this is a team effort. We have many collaborators involving data provide and generations, and our manuscript is available. So anyone's interested in, you can look at our recently published manuscript, and we thank for our founders. So our Genomics to Proteins portal is rooted based on this central dogma of the biology, where the genetic information flows through DNA to RNA and proteins. So one can identify genomic variation in their genomic coordinate, can translate into this cDNA, which is the coding DNA consequences, and eventually amino acid change. So this is a holistic understanding of how this information flow from DNA to RNA to protein can help to build our biological hypothesis on disease mechanism and further help to develop therapeutic strategies. So at this point, now you're wondering, why would you need a specialized portal for this? And the answer lies to this unprecedented surge in biological data recently. So first, this genetic variation. Thanks to the high throughput screening, we can now identify very, very large number of variants within the human populations and disease-associated groups. And especially if you look at the number of genetic variation found in people as time goes on, you can see that this variance of uncertain significance grows really fast. And in 2025, it reaches up to 1 million variants that we don't know the consequences of it. And there's also large advance in the functional genomics, where you can now base edit with the CRISPR-Cas types of study, that you can study demutational scanning, where you can identify this functional impact of the protein in a very deep way. And also there's a large number of structure of the protein available. So experimentally, we have a protein data bank where it contains very large number of experimentally validated protein structure. But recently, we now have AlphaFold, so we can generate almost every protein structure available, reaches up to 200 million predicted structure. So the question at this point is, how can we usefully integrate this data and scale up our investigation? This integration is particularly useful, especially in the disease context. So you can look at this DDX6 RNA helicase example, and researchers can identify de novo variants from patients. suffering neurodevelopmental disorder. And if you map those de novo mutations into the protein structure, you can clearly see that those mutations are actually located very closely in the RNA binding motif, and it affects its functions by this mutation. And this is not only the DDX6 case, and we can find many examples where a disease mutation clustered in the particular region of the protein's 3D structures, and in certain cases, these different clusters in their 3D structure connect to different phenotypes of the disease and severity. So we can see this GABA receptor case. There's a cluster where it produced mild epilepsy, and in other cluster it produces severe epilepsy. So for in this portal, we try to streamline this effort to integrate information from genomics to transcriptomics, proteomics to structural biology. So in the portal, if you can search by gene, you can see many different transcript identifier from Ensemble and the VICC IDs, and we connect it to the UniProt protein identifier for all canonical isoform. Then we can also connect it to every available structure from the experimental databases and also predicted AlphaFold structure. So with this integration of this identifier mapping, we can map more than 49 million genetic variations found in the ClinVar and HGMD and NOMAD to more than 88,000 protein structures to the PDB and AlphaFold databases. So let's look at what's the genetic variation in our portal. So by those structure mapping, we can map over 90% of variants on canonical protein isoform mapped on these protein structures. And we integrate ClinVar, HGMD, and NOMAD, and we offer also database-specific filters. So you can see ClinVar variation by their molecular consequences and also pathogenicity. And for HGMD, we also have molecular consequences and this disease mutation confidence. And NOMAD, you can search variation by their allele count and allele frequency as well. So, and this genetic variation direct to the protein structures and protein sequences. So we integrated and investigated multiple databases regarding the protein space, and you can see multiple tracks of protein features. So I'll briefly introduce what's available in this protein structure and feature space. And one is physical chemical property. This is about the side chain property and chemistry of it. And you can also see the structural features, such as the secondary structure of the protein, solvent accessibility, and we also provide this AlphaFold confidence because not every variant is located in this highly confident AlphaFold structures. And we also provide this pocket detection where is the drug binding pocket sites. And the other one is the protein interaction features, where the hydrogen bond, salt bridges, disulfide bond, van der Waals interaction. So any variant is involving in one of these protein interactions. And we also have the protein sequence annotation from the UniProt. It has many different 32 annotations, including some of the functional sites and domain informations. So for example, you can see available annotations such as this receptor, lipid binding receptor, like EGF-like domain, and many information available in the UniProt. We can integrate it with your variant annotation. And also post-translational modification is available, including acetylation, methylations, and this is collected from this PostSite Plus database. And also, whenever available, there is a database called MAVE, which is a public repository for data sets from multiplex assay of the variant effect. So this is a combination of literature that they investigate this functional deep mutational scanning study. So there are currently 500 proteins available in their MAVE scores. So our portal has two different modules. So first module is the research module. This allows to look up your gene or protein, and you can see this complex integration of the databases. So for example, if you look up your gene MORC2, you can select your relevant transcript. Then you can see all of these protein features that I just explained in a single track. And then you can map those protein features and variant at the same time into your structure viewer so that you can map your ClinVar pathogenic and likely pathogenic missense variants along with other protein features, such as in this figure, it shows this binding site, so you can see how they are located in their protein structure. And the second module is the interactive module. And in this interactive module, you can not only look up your gene and see the available resources, you can also upload your own protein structure if you have your unpublished structure. And also you can upload your customized annotations like screening outputs or your own clinical annotations, etc. So you can upload your scores, and now you can see these tracks as well and your input structures there, and you can interactively work with your data and the currently available data. So what's important is, because clinical data is really time sensitive, we provide this biannual updates. So we do the major update of this protein structure and feature and this ensemble the databases biannually, and also this ClinVar is really updated in a frequent basis. We update this ClinVar monthly basis, and this is our the most recent update of the September 2025, and you can also see the log release of our portal data. And also those who work on this bulk data, you can use our API access for automated data retrieval analysis. And we have repositories as well. So anyone who codes or do the machine learning, you can use the bulk data download from our API. Since we launched 2022, we have more than 10,000 users across the world. That shows that the community really needs that types of tools, and the GTP portal is filling up this unmet capability. And what's coming up next is the protein feature enrichment score. This is kind of our interpretation, because we are building and collecting protein features in a whole protein-wide scale. We can now characterize like which protein features are actually enriched in the pathogenic variation compared to the population variant. So we are curating our data sets, our controls and cases, and we calculate the effect sizes of each features available in the G2P portal, and we can compute the score out of this protein structure and sequences, and so that you can have this in silico interpretation score based on the protein features by in a like deep mutational scanning way. And then what's important is that because we are collecting protein features in many different categories, we now can decompose the score into different molecular mechanisms, so that if you have a score, you can see actually, and you can interpret the score based on these different categories. So if I give you example, this PPAR gamma gene, this arginine 194 tryptophan is a known pathogenic mutations relevant to this lipodystrophy. And if you look up this mutation and its protein feature enrichment score, it has a high score, and we can also provide this posterior probability of pathogenicity. So it's interpreted as being highly pathogenic, which is true. And we can now decompose this score, like where this score is coming from. And it's coming from some biochemical property change, and it's coming from some structural stability change because it's located in the protein core, and it's well folded, and it has hydrogen bond and this really high confident alpha fold regions. And it also contains some functional sites relevant to the pathogenicity. So it has DNA binding regions and zinc finger. So not only interpreting its pathogenicity, it shows like which feature is actually contributing to its pathogenicity. And we can see these types of heat map where you have a high like signals on those regions in their functions, because it has DNA binding sites. It has some like domains and structural features as well. So this score will be coming up in the G2P portal, so you can so helps to interpret your variants in a protein feature-based way. So we can always welcome feedback. So if you have any feedback, you can use our tab, and we have YouTube tutorials. So you can see our tutorials whenever possible. And we yeah, so Sumaiya's gonna showcase the portal. So yeah, stay tuned.
All right, can everyone hear me?
Yes, I can hear you.
Awesome. This is not connected to the audio, right? It should be the entire room.
Yeah,
okay, awesome. All right. Okay, so in the next part, what I'm going to do is I'm going to do the live demo of the portal. But before that, my name is Sumaiya. As Beth introduced, I lead the Bioinformatics and Machine Learning group at the Ladders to Cures Rare Disease Accelerator Center at the Broad Institute, and I'm very happy to be here today and showing you how to use the portal. So please feel free to ask me questions as I go through it. Seulki just mentioned that we have 2 different modules in the portal. Let's see, where I have it. Okay. Okay. So I hope everyone can now see the portal homepage. So the 2 modules that Seulki just described was the resource module and the interactive mapping module. So the resource module is basically meant to query by gene name and then see and visualize the mutation information, protein information, and then get a summary interpretation, which I'm going to show you in details. And then the interactive mapping or interactive module is meant for the users who instead not like to investigate only the data that are available in CleanVar or Nomad, but also would be interested in uploading their own mutation and then map it to the structure for analysis. So I'm just going to stick to one example that I can use through displaying the resource module. So for that, I'm going to switch to another case study, but I'll for now just look up TP53. And here, after you enter the gene name, then you land onto this page where first you will see some generic information for the gene. And also because this particular portal we developed focusing on Mendelian genetics, we have now fetched any rare diseases that are associated with this particular gene, which is basically what you can see here. I think you all know this database, which is GenCC database. So in this gene page or under the resource module, the first page that you can visualize, we have these 5 different tabs. In the first tab, you will see all the protein feature annotation residue-wise that we have brought together in this resource module, and then the variant data, and then the variant to protein structure data. And finally here, this is basically the backbone of the portal, and it is important that I discuss this particular tab, which is a mapping between the UniProt identifier, which is the isoforms, to different transcript identifiers. And the reason this is the backbone of the portal is because many times variants are annotated in non-canonical transcript, and I will showcase one of the examples. And what we need to do to tackle those situations is we need to do the sequence alignment between non-canonical isoform to the canonical isoform, and then map the variants over to canonical isoform. And so this table basically shows you that, okay, for this particular isoform, we have these transcripts that are canonical, but there are many other transcripts actually that encodes the same protein. And there are many others that encodes different protein. I mean, TP53 is a complex protein. So for this, we have a lot of isoforms. So I'll go back to this protein sequence annotation. And the first thing that I would like to highlight is this isoform alignment feature, right? So let's say, right, by default, it will show you the default isoform, but you can click on these other isoforms and can see that what part of the protein is basically missing. So variants from that particular part for that protein will not be mappable to the canonical isoform, obviously, but this type of isoform alignment also gives us other hints. For example, what are the domain or the signal peptides or the motifs might be missing from this particular isoform, and what could be the functional consequence of that missing region of that particular protein. So there could be cases where we have a non-canonical isoform that is shorter than the canonical isoform, and for that case, it will show you that there is an alignment gap in the isoform sequence, but it could be opposite as well. We have non-canonical isoforms that are longer than the canonical isoform, and then it will show you the breakpoint, and it will tell you that you have a gap in the canonical sequence. Now, moving on from the isoform alignment feature, as Seulki was describing, that we have a whole suite of protein features annotated residue-wise that we have brought together, including the physicochemical properties, the structural features such as secondary structure, accessible surface area. So I can click some features on, and you can see that this is an interesting region of the protein, because if I, in the UniProt sequence, turn on the DNA binding domain, then this is the DNA binding domain. And there are lots of region annotation. Many of them are disordered region, basically. But what is interesting is this DNA binding domain part of the protein is very structured. As you can see from the AlphaFold prediction score, this blue indicates that the prediction of AlphaFold is very high for that particular region of the protein. And also, this is the particular region of that protein where the accessible surface area is very low, meaning the protein is mostly is folded and forming, and most of the residues are at the core with lower accessibility and having a high AlphaFold confidence, and which is also a modular domain, which is a DNA binding domain. And this nicely aligns with the MAVE score that are available. Actually, for TP53, we have a lot of MAVE data available. And you can see all the proteins for which MAVE data are available from the statistics page of the portal. Lots of pop-ups. Okay, so in the stat page, you can go to the end and then the multiplexed assess of variant effect data. If you click here, it's going to take some time to load, but then you can find what are the proteins for which MAVE data are available. Coming back to where we were, what I was showing is this presence of the structure domain, DNA binding domain, and this red part of the MAVE score nicely aligns and shows you that this is the region that is particularly important for the function of the protein. Couple of other important information is you can click on somewhere on this feature viewer, and it will show you the residue properties. Will give you a summary of the all the features that you can view, also in the text format. For example, I clicked on the residue position 102, and it gives exact feature values, the protein-protein interaction features, the UniProt features, the post-translational modifications, and also the variant effects for that particular position. You can zoom into the viewer to find out the exact residue position and then click in there to find the protein features of that particular residue position. Moving on to the second tab, variant to protein sequence, here you can see all the features that were there already on the feature viewer, but it will bring in now the variant database, the data from the variant databases. So that includes Nomad, so you can see different variation type, missense synonymous, and also can filter them by their allele count and allele frequency. So I'm going to deselect Nomad data from the viewer, and then we'll focus on the CleanVar data. So here we have the missense mutations mapped, and the pathogenicity filter is on, which showcase, and also the AGMD mutation data are also available in the portal. I don't think we have a filter applied here, so I'm just going to apply the filter. So we have more data from AGMD, but in general, AGMD data are less reliable than CleanVar data. So I'll probably just take it off, and I'll take it from the left side menu as well. So now what you can see is this CleanVar data are also, the pathogenic mutations are also nicely overlapping with this DNA binding domain, and also with that same regions where we had different variant effect compared to other part of the region. So looking at this scores, it's pretty clear that this is the region that is highlighted in that MAVE, and that nicely overlaps with the DNA binding domain, the AlphaFold high prediction, and also the CleanVar data. So next I will move on to, okay, so before I move on to the variant to protein structure, probably it's important to showcase that this particular set of variants are coming from these transcripts, but you can also select another transcript if that is the transcript of interest and map that data over to the canonical sequence. So I'm not sure for this transcript if the data are available, but let's see. If I select this other transcript, then what it shows is no CleanVar data are available for this transcript, but Nomad data are available, so it's showing the Nomad data. So this is the feature to align the variant data from a non-canonical isoform to the canonical isoform for the interpretation. And when we have the variant data mapped in the same way, you can click on the variation information, and now you can see side by side for that particular position that you clicked on, the variant data in details from CleanVar with the clinical significance and phenotypes as available, and also the protein features on the side. Now moving on to variant to protein structure. We have now the list of experimentally solved structures that are available. Sometimes it is difficult to choose which structure to select from. One thing sometimes I do is I kind of apply this filter to see what are the structures where we have more residue coverage. So here, if I sort in the decreasing order, then we have this one with the highest coverage. And something you are noticing here that the portal is failing to bring in AlphaFold structures, which is because AlphaFold just recently updated their database, and then they added the version 6 data and replaced the version 4. So we are still working on updating the portal. And meanwhile, we are showing the first version of AlphaFold. So this is something that happens all the time with an online, like live dynamic portal. So we always keep on, at least we give our best effort to update the portal as other databases update their data. So now, because AlphaFold is not available, we have experimental structure. So I can click on this structure with the highest coverage of the TP53 protein. Now you can see that this is the structure, which is very big, but it is a very interesting structure, and it has a lot of chains, as you can see, and a lot of other proteins are involved in this one structure. And this colored part will be the target gene of interest. So basically the colored part of the structure is the TP53. So what I'm going to do here is I will change the layout, and I'll take the structure here just so that the structure is little big. And now I will go up and will highlight the DNA binding domain. So you can, this sequence viewer and structure viewer are coupled, so they can talk with each other. So if you select on the row header, this feature is mapped onto the structure and we'll see it. So I'm mapping now the DNA binding domain, and on top of that, missense mutations. So now if we scroll down, so let's take off the missense mutation to begin with, and let's see the DNA binding domain. So this is this green color part of the structure is the DNA binding domain of the protein. And if we map the missense mutation, then there is a lot of missense mutations on that particular domain. And interestingly, not in the other part of the protein, only on that domain. So, and here you can, there is a lot of data. So I'm seeing some flickering going on, but you can hover over on specific mutations to see the disease information and also the protein consequences. Note that you can also download the data in tabular format if you wish to see in tabular format. So for example, I can click on protein features, and I can from here also go to CleanVar variants, and here you will see the data in the tabular format. And you can filter the data and sort the data in different manner, and also the protein features that are sorted residue-wise. You can select or deselect different features that you want to analyze, and can also download the data. So this sums up my demonstration of the resource module. I'll move on to interactive mapping module. And if there is time, I'll come back and we'll showcase an example that I thought that I'll showcase. But even if I cannot show that, I can actually share this slide deck with you, which is analyzing the molecular level effect of a variance of uncertain significance in the KIF2A. The reason I find this case study very interesting is because this particular variance of uncertain significance is also on a transcript that is non-canonical. So this particular example really shows you that how you can, so the steps are written, as I said, I can share it, how you can use the portal to map it and identify that how that variance of uncertain significance, although it's a VUS, by structure analysis, you can clearly see that it would break an inter-protein interaction, and also it is on the functional domain of the protein. So if we consider the molecular level effect of that particular mutation, by our analysis, we would definitely not categorize it as variance of uncertain significance, but we will categorize it to have a molecular effect on the function of the protein. So I can share this example. So I'll probably pause here for 1 second if there is any question. If not, I'll move on to the next part, which is the interactive module.
Thank you so much. Yeah, I have one question.
Yeah.
A lot of times when I'm looking at a variant, I have to kind of convert a C dot or P dot between like different isoforms of the protein. Is there an easy way to do that using this tool?
I mean, if you have a C dot, then you need to convert it to P dot. That I understand, but
I'm thinking like C dot from one transcript in the same genomic position, and then like converting to a different.
So now this actually tells me that probably it makes sense to spend 2 minutes on this particular example. So let's say, because this is that example that will show you how to map variant from another transcript. So let's do this. So what I'm going to do is KIF 280. So here, okay, let's look at the case study. So the case study is we are looking at glycine 478 arginine, and this is the transcript glycine 478 now. So this is by default the transcript. We'll go here and see what happens. So when we go to variant to protein sequence, this is the -1 that is selected, but the canonical protein isoform is -3. So all the protein features that are available are for -3, right? But by default, this is selected. So what we will do is first we will turn on the -3. But as we open this list, I think an important information to notice is this is the canonical protein isoform that is this asterisk shows you, but it is not associated or mapped to transcripts that are also canonical. So the transcripts that are canonical are here. So this is a perfect case study where the canonical transcripts on which most of the variants are annotated at are not mapped to the canonical protein isoform. So we cannot do molecular level interpretation instead other than transferring the variant. So let's first select the canonical protein isoform. So this is the isoform, and our case study was glycine 478, and it's a VUS. So what we have to do is, okay, we selected the canonical isoform, and the CleanVar shows that there is no CleanVar data available. So we cannot map CleanVar VUS, right? But we can. That is, we have to click here, align variants from another transcript. And now we can select this one where CleanVar data are available. So I'm going to select this. Now see what happens is, now this missense mutations from CleanVar is from a non-canonical isoform, which is now aligned onto the canonical isoform. Now remember that by default, we always show the pathogenic mutation. Now if we keep this selection, then we will not be able to find our 478 VUS. So we need to select the VUS. Now, let's find our mutation, glycine 478. I'm trying to expand this viewer, which is not working. And I'm going to refresh this. Align variance from this one. So this is the position, glycine 478, and this is the VUS. So glycine 478 position has 2 mutations. So I'm going to click on there, and the protein consequence, the first one that is available is 478 to alanine, and then 478 to arginine. And this is the variance of uncertain significance. It has a phenotype associated, but definitely not enough evidence to categorize it as pathogenic. And when we look at the protein features, we can see that this change is substantial change in the physicochemical properties. It is located at a highly folded region of the protein and also breaks some protein-protein interaction. It's on a domain. And by structure mapping, we can also show that how it is on the protein-protein interaction interface. And from this summary, you can take a note that to see that protein-protein interaction effect, we have to select this particular structure 6BBN, right? So I'll go to variant to protein structure now, and let's find this structure 6BBN. So now I'll go here, and I need to do the same thing to find the variant, which is align the variant from this particular transcript. And I will repeat that exercise. It seems like it's showing all the variants, but I'll go with the VUS. And in the same way, I'm going to expand here to find the mutation here. I'm going to map it to the structure. So this would be that protein-protein interaction interface. This is 470. And I'm basically now looking for the 478 position. The better way to do is basically I'm going to change the layout and bring it side by side. And when I hover over, I can see that mutation position exactly right here.
Wow.
The 478 position, this one, that it clearly shows that how it's within the protein-protein interface. Now, one challenge I definitely recognize is that without a structural biology opinion or understanding, it is very hard to interpret it looking at the structure. And that is why I think the most important or the convenient ways to look at the summary interpretation that features and see what are the features that it is breaking. But to make it more intuitive, what we are developing is that score that Seulki was showing. And that is the score that will give a quantitative value showing that, okay, with this confidence, we can tell that this will have an effect on protein-protein interaction. But Beth, I hope that with this, it is at least clear that when you have a variant annotated on a different protein isoform, how to take it to the canonical isoform.
That's perfect. Thank you.
Awesome. So with that, I'll move on to this next module, which is the interactive mapping module. Here you will need to sign in with your Gmail address. And this is just to keep your data secure because people will upload their own structures that might be unpublished, and also their customized mutation data that also could be unpublished. Especially this is the case with the base editor tiling screening data, when scientists want to understand the molecular effect of their unpublished data, then they can upload it through here. So to showcase this one, I will go ahead and we'll start with the gene. And here I'm just going to showcase this example that I have it ready here. And I will also share this particular case study with you, where I will focus on this particular gene and some resistant mutations that are known. So first you type in your gene name and then proceed to the next step, where you will need to select your structure. And the structure of interest for this particular case study is this. So we will analyze this covalent resistant mutations, and then our resistant mutation to the covalent inhibitor, and also some acquired mutations to the non-covalent inhibitor that were all analyzed onto this particular structure, which is 5P9G in that particular publication. So this one. So after I select this structure, it loads here, and then I'll go next to the annotation. And here, what I'm going to do is, this is the annotation uploader, and it is important to know how to use it. So you can paste your CSV or TSV data directly, and you can also upload your data. So if I go ahead and upload my data, then it will parse your data, and then will show you whether you have uploaded mutation data or feature data or score data, because we allow 3 different types of data. So I'll go ahead with the paste data, and then we'll go ahead and upload the default mutations to begin with before I upload the customized mutation from the paper. So when I submit these annotations, see, it automatically categorizes this data into mutation based on this format. But if it is the wrong format, then you can also change it to feature or score, because it can do mistakes sometimes. If you want to filter out, let's say, one column, then you can just filter out that particular data that you have uploaded, and then submit your annotation. When you do this, this is your own uploaded data, and you can map these mutations here. These are just default data, right? It has no significance. One important information is all the features, protein features that you can use for interpretation that are available in the resource module, you can also bring in the interactive module. So these are the nomad data, existing ClinVar data, and all the protein features. So this is for you to analyze whether the mutation you just uploaded, whether it is very close to other ClinVar mutations. Sometimes this one piece of data, that how close this new de novo mutation that we have that we do not know anything about is how close it is to the other known pathogenic mutations. So this feature is to enable users to do that kind of analysis. So here, what I'm going to do is I'm going to go back to my annotation uploader, and I'm going to go back, and here I'm going to upload this one additional piece of data, which is my C481S. So C481S, right? And I'm going to submit this, submit annotation. Oh, okay. So there are 3 columns, but I have uploaded only one piece of data, so it's showing me an error, which is very legitimate. So what I'm going to do is I'm going to add 2 commas. Hopefully that solves it. Valid mutation at position which I'm not sure about. So C481 because it's on C482. No, this is the C481. This should be right. C481. Yeah, okay. So now I submit this annotation. So this is my C41. So this is not benign, but I did not change the column name, so it doesn't matter. So now these 2 were those default mutations with no significance, and this is the mutation that I just uploaded, right? So this is, remember, this is a mutation to which is a resistant mutation to this covalent inhibitor here. So what is immediately noticeable that why this mutation would be a resistant mutation, because it is directly on this inhibitor binding site. So this is the type of mechanistic understanding at the molecular level that we hope that this portal will enable general community to investigate and understand. And I will share this particular case study as well. This is just a part of the case study, but the full case study shows not only why these mutations are resistant, this particular mutation is resistant to this covalent inhibitor, but there is a set of somatic mutations that are known to cause resistance to the non-covalent inhibitor as well. And if you map this mutation on the structures, then you will be able to see that why these mutations might also cause resistance. So with that, I'll probably stop here with one additional piece of information, which I just remembered that it would be important. So when you investigate the mechanism of resistance, another piece of data that is available in the portal could be very useful is the pocket information. So the drug binding pockets that are annotated already, which is here under structural features, the pocket number and the druggability score. So I'm now bringing in that data, and this is our structure coverage, and I will map this pocket data here. And after you map the pocket data, it is even more clear that these residues that are cyan color here are basically forming this drug binding pocket where these mutations are. And this data will be particularly useful if you carry out that second part of this case study where we map the somatic mutations to the non-covalent inhibitor. And you will see that although a mutation may not be directly on the ligand binding site or drug binding site, if it is within that pocket, then it still has a resistant effect. And that was about it. So I'll stop here. I hope this is useful, and we are open to question. And I think most importantly, on the portal, on the top right side, you will notice a feedback button. And if you have any feedback for us, please don't hesitate to reach out to us, and we'll try to address your feedback. So I think that was it. Thank you all for listening.
Great, thank you so much. That was so nice, so nicely done. And this recording will be placed on our YouTube channel as well, so we can reference this and have ease of accessibility for all of our members and surrounding community. So thank you so much. This was really great. Are there any questions from the audience today?
So I have one question. Is this related to only missense variants, how you're dealing with the indels in those structural model and how those are highlighting those kind of situation?
It's a great question. So we do map indels and other variations anchored on the beginning at the protein level where the change starts to occur. But that is, so what you have to, the way you have to extrapolate the molecular effect is from where the beginning of the effect is, you will miss the rest of the protein's features and function, basically. But other than that, it is about the missense mutations, because at least you have the full protein and there is only one change, and you can understand the effect at the protein level or the protein feature level.
And as I heard, if I hear correctly, you are trying to give some scoring system. Is there any scoring throughout the protein domain structures which you are providing with the cutoff where, like for us who are in the clinical labs, who are at a glance can see and predict that if a variant falls in that domain, in that, and it's go in the pathogenicity level from higher to lower end?
Yeah, that is exactly what is coming.
Yeah.
That is so first challenge was to get over with all this transcript misalignment and mapping onto the structure. Experimental structure data was extremely messy. And now we are working on actually 3 different things. One is quantitatively allow the users to identify whether a new mutation is part of a cluster of already known pathogenic mutation. That is a quantitative score that is coming. The second is the protein feature score that is coming. So it will really show you that, okay, this is probably the score 10. And of that, 8 is coming because it is on a domain that is functionally so important. So it's going to have an effect on the function of the domain. And the third interesting thing that is coming, we are yet to see how useful that will be, and that is conversational artificial intelligence agent that will be able to talk to people of different background and give and query the data that we have put together and give you a summary result. So these all things are new features that are coming, and I will be happy to give another session sometime next year to describe all that.
One follow-up question on that. Are you still, this model is also capturing the splice variants?
No, we are not capturing splice variants because our interpretation is with protein features and with protein structures. So this is kind of a little bit out of scope for the evidences that we are considering in this interpretation.
Thank you very much.
Great. With that, we're a little bit over time, but I just want to thank you again, Seulki and Sumaiya, for coming on and giving us a tour of G2P portal. And thanks for answering all of our questions. And I'm excited to see where this tool goes in the future. We'll be in touch.
Or Beth, would it be helpful if we share our slides, or that is not necessary because we have recorded the session?
That would be great. I can put it on our Google Drive and share it with our team.
Awesome. Thank you. Thank you, everyone.
Thank you.
Bye.
Thank you all. Bye-bye.